repute. He also has published more than 50 popular articles for the benefit of the farmers on improved technologies in the field of plant protection. Prolific writer, Dr. Rai has authored 16 books and has contributed more than 30 chapters in the volume published by national and international publishers. He has been awarded the Rajbhasha Award in the year of 2013 by the Honorable President of India for his contribution in scientific writing in Hindi. Apart from this, Dr. Rai has also been the recipient of the Scientist of the Year Award 2015, Award of Excellence for Energy Awareness in Agriculture from IIT BHU, Lifetime Achievement Award, Best Paper Award, and many others. He has guided both the MSc and MSc dissertation and PhD thesis of 7 students. He is on the panel of examiners of several academic institutions and on the editorial board of some of the prestigious journals, including the International Journal of Agricultural Sciences. He has served as the chairman, member of various committees, and is on the board of studies of Mahatma Gandhi Kashi Vidyapith, Varanasi, Uttar Pradesh. Dr. Rai has also served as resource person for training lectures sponsored by USAID, which was working in our BHU, National Center of Organic Farming, National Horticulture Research Development Board, IIBR Varanasi, Department of Agriculture, Government of Uttar Pradesh, Ministry of Earth Science, Government of India, and Skill ASCI, Government of India, Rashtriya Krishi Vikas Yojana, and number of other reputed organizations. So I, on the behalf of organizing committee and on my personal behalf, I welcome Dr. J.P. Rai sir. So kindly giving your shower of knowledge to our participants. Sir, my PPT is visible or not?
Yes, sir. PPT is visible. Please go on slideshow mode.
Yeah.
Okay, sir. Fine. So go to the next slide. JP, sir, please start. JP, sir. Hello.
Yes, am I audible?
Yes, sir.
Hello,
Yes, sir. Please start.
am I audible?
Yes, sir. Please start.
Okay.
Yes,
So
sir.
the agenda. Okay. Thank you. So the agenda for today's discussion will be seed health, testing for seed health and technologies associated with it, the objectives of seed health testing, the methods that are utilized to test the health of seeds, protection of seeds, the challenges and prospects ahead, and lastly we will summarize it. Next, please. Dr. Yes. So seed health, this is the definition of seed health. It refers to the presence or absence of disease-causing organisms, including fungi, nematodes, bacteria, viruses, and insects, of course, insect pests, and to the status of seeds in a seed lot. Next, please. Next, Dr. Goel. Yes. So it has a lot of significance. These are some seed trade associations, including India. So there are 2 seed trade associations of India, Federation of Seed Industry of India, as you can see here, and NSA to the middle right, National Seed Association of India. One more association, IS, stands for International Seed Testing Association. It is in fact Irish Seed Trade Association. So there are various seed trade associations across the world that need healthy seed, and seed health testing is done for them. Next, please. Seed health testing is, of course, a prerequisite for seed trade. The objectives of seed health testing is that seeds need to be protected from a number of biotic and abiotic factors that affect their quality and their performance. And seed health testing is also the prerequisite of designing the seed production strategy. It's also done to know the planting value of given seed lot in order to forecast the field emergence, plant stand, and predict the health of the mature crop, along with forecasting of the benefit cost ratio of the crop and its economy. It is also necessary to know the storage quality or feeding value of a seed lot. Seed health testing is also done to check the advisability of seed treatment. It is also done to know the efficacy of seed treating chemicals. Next, please. Okay. The basic requirements of seed health testing include that it must be efficient with optimum level, desired level of accuracy and precision. So what desired level is that? It relates with the economy and the amount of precision. The test must be simple and it must give reliable information pertaining to the feed performance and quarantine requirements of the seeds. Now, the results that are obtained by these methods, they must be reproducible within the statistical limits. The time, labor, and equipment for carrying through a test, these must be kept within the economic limits. Very expensive testing method will add to the cost of the seed, and that will leave an adverse effect on seed trade. Without compromising the sensitivity of the test, of course, the sensitivity must never be compromised. Seed health testing. Next is the test requiring incubation must be able to give the results quickly, because that is what it is meant for. Next. So the methods that include, we will take an overview of them. Dry inspection and visual observation is the most common method of seed health testing, and it is done by an expert, or even a farmer can do that and get expertise in that. So what we look for in visual observation, number one is inert matter. The inert matter admixed with the seed includes plant debris, spotted, unfeed and chaffy grains, sclerotia of various fungi pathogenic, galls of nematodes, smut balls, and insect pests and their remnants. Now, it should also be incubated either in blotter paper or on agar media and examined after a standard period of infection. Next. So dry symptoms, we look for symptoms while visual observation and dry inspection. We look for symptoms such as discoloration, staining, necrosis, malformation and similar indications of infections, fruiting body of fungi, resting hyphae on seed surface, spores or bacterial masses on the seed, as well as mechanical damage. These are the things that we look for in visual observation and dry inspection, and we can deduce our observations based on that. Next. Again, advantages and usefulness of dry inspection, that it is pretty much economic. It provides quick information and does not require much equipment, and it helps to take the first hand measure. The limitations of dry inspection is that only those diseases or admixtures which are external to the seed, and symptoms which are external and are manifested on the seed coat, and signs of the external to the seed can be detected by this method. Internally seed-borne pathogens and organisms cannot be detected by visual observation or dry inspection. Information pertaining to the seed viability cannot be obtained by visual observation or dry inspection. A seed looking pretty healthy may not germinate. It is not more reliable because in badly infected seeds, I told you that badly infected seeds, they may look pretty healthy, but upon germination, it is found that they could not germinate. So their quality is compromised. Next. These are the equipments that we need for visual observation or dry inspection. We look for admixtures, discoloration, malformation, necrosis, fruiting bodies, abnormality in size and shape, that is shriveling, fluorescence under near ultraviolet, radiation. For example, greenish wheat, if you see, we suspect that it could be infected by Septoria nodorum. White or cream bean, if you see, we can suspect that Xanthomonas campestris pathovar phaseoli might be infecting the seed. Bluish white bean, it can indicate that the infection could be of that of Pseudomonas syringae pathovar phaseolicola. Next. Discolored and spotted rice grains, they can give the indication that it could be due to Bipolaris oryzae, Pyrocularia oryzae, Carvularia lunata, Cladosporium oryzae, Nigrospora oryzae. So these give primary indication upon visual observation or dry inspection. And big species may also be admixed with the seeds that come from the threshing floor during threshing. Next, please. Visual observation or dry inspection gives us an indication of black point of wheat. The embryonic point is black or dark. The main causal organism is associated with it is Bipolaris sorokiniana. Some other organisms that may be associated as saprophytes include Alternaria tenui, Curvularia species, Fusarium species. Next. Dried and discolored and sugared lightweight jute seeds. They can be indication of association of Microphomina phaseolina, Botulidiplodia theobromai, Colotrichum carcori. Next. Of mustard, sugared and discolored seeds of mustard may give an indication of the infection by Alternaria brassicae, Alternaria brassicicola, Aspergillus species, Penicillium species, Pomalingam. Next, please. Purple stain of soybean. It is a rather pretty sure method of inspection, dry inspection. By dry inspection and visual observation, we can be pretty sure that the disease is there. Seeds are infected with purple stain because it is so conspicuous. The seeds are purple in color at one side, and other side remains normal. The causal organism is Cercospora kikuchi. Sweet gourd, discoloration of sweet gourd seeds may indicate that Colotrichum and Aspergillus and Alternaria species may be associated with it. Next, please. Okra seeds, when found discolored and sugared, it is suspected that Colotrichum dimycium, Microphomina phaseolina, Aspergillus species, Penicillium species, and Fusarium species might be associated with the seeds, and the seeds might be infected. Next, please. Discolored and sugared seeds of country bean. These are the pathogens that have been listed. Next, please. Mung bean. In mung bean, if we find discolored and sugared seeds, we may suspect that list down to 8 pathogens: Colotrichum dimycium fungus, Cercospora canescens fungus, Microphomina phaseolina fungus, Botulidiplodia theobromai fungus, Fusarium species, and Phoma species fungus, and mung bean mosaic virus and mung bean golden mosaic virus. These 2 viruses may also be associated with it. It is the primary suspicion, and we go for the further test to confirm. Next, please. Discolored seeds of white gourd, Colotrichum, Aspergillus, and Alternaria species may have been associated with it. Next, please. Now we are coming with the advanced level of visual examination, that is microscopic examination. In microscopic examination, there are 2 methods mainly. First is washing test. Washing test is done to collect the propagules, pathogenic propagules or fungal propagules present on the surfaces of the seeds. What we do, that measured seed quantity with fluid, that fluid might be water or alcohol, it is washed through mechanical checker, and measured quantity of this washet is examined under a microscope for presence of pathogen. For example, if you take 1 drop, it is approximately 0.05 ml or 50 microliter. So the propagules found, it is counted, number of pathogenic structures per unit of seed weight. It is calculated based on the count of the propagules that we find in the microscope. Viability of pathogens may be ascertained by plating a measured volume of fluid. Now, when we want to test the viability, we should refrain from washing it with alcohol, because alcohol kills all the propagules, pathogenic propagules, and sterilizes the seed. So wherever viability of pathogens is to be recorded, the washing with alcohol may be avoided. Now, next is whole embryo count method. The protocol for it is given in the further slides. Next, please. This is the protocol for whole embryo count method for testing Next, Dr. Goel. Yes, this we can see in observation, microscopic observation, that this embryo is the first one is healthy embryo. We do not see any fungal mycelium or mycelial growth. Whereas in this second picture, we can see that the dormant mycelium is all surrounding the embryo. Next, please. This is the dormant mycelium that we are seeing in dark color. Next, please. So these are the fluorescence that we see. This is in fact the mycelium of the fungus growing with the apical point of the seedling. Next, please. Yes, hyphae that are being seen in fluorescence. Next, please. Again, next. let me see. Let me show you that this is the invasion of fungi by on Primordium. Next, please. So these are beautiful pictures that that can be seen, and we can be assured that the seed is infected or not. Next, please. Cross section and lateral section of the node. When we cut the plant, cut the transverse section of plant, we can see that the fungus is present all over there. Next, please. So now incubation tests. Incubation tests are rather confirmatory, but if they take more time. These involve standard moist blotter method, SMB method, earthen dish newsprint method, agar plate method, deep freezing blotter method, and 2,4-D method. Next, please. We'll discuss one by one. SMB layering of sterilized blotters in petri dishes, 1 to 3 layers, and buffer soaked absorbing paper, and moistening it with water, and placing of 10 to 20 non-sterilized seeds, depending on the seed size, in one petri dish, and incubating it with 12 hours of light and darkness for 7 to 10 days at 25 plus minus 2 degrees Celsius. After the incubation period is over, seeds are examined under a stereo microscope for presence of fungal colonies, and their characteristics are recorded for the identification of the fungal pathogens. The germination of seeds may be obstructed because when seed will germinate, their germ fluid may be colonized by some external contaminants. So germination may be restricted by wetting the blotting paper by with 0.1 to 0.2% 2,4-D. That is a herbicide. Next, please. Yes, advantages are that the pathogen can be detected quickly by observing their growth characteristics. It's economic. And most importantly, that the pathogen that we do not, slow growing pathogens that are not visible in agar plate method or other advanced methods, they can be detected in this because there is no nutrient and only humidity is there. So the growth of all kinds of pathogens is slowed, and the fast growing pathogens, they do not invade the whole petri plate, and the slow growing ones, they can also be detected. Next, please. The limitations are that it may be hampered due to fast growth of some fungi on the blotter, which are cellulose consuming one. Pathogenic bacteria, they cannot be detected by this method. It is time consuming. The pathogenicity cannot be detected. Symptoms may not be detected. Next, please. Next. Now, earthen dish newsprint method. This is an indigenous method of standard blotter method. So it is very inexpensive. Petri plates are replaced by earthen dish, and with earthen cover, earthen lids, and blotter papers, they are replaced by newspapers, and the process remains the same. After incubation, we can see the growth of fungi and pathogens present on and in the seed. Next, please. Agar plate method, rather defined method, and it needs some equipments like autoclave and incubator, laminar flow, etc. So seeds are first seeds are subjected to surface sterilization by 0.1% solution of mercury chloride or sodium hypochlorite. After 3 successive washing with sterilized water, these seeds are placed in petri dishes that contain PDA, potato dextrose agar medium, and incubated for 5 to 7 days at 22 to 25 degrees Celsius under 12 hours of alternating cycles of light and darkness. 5 to 10 seeds are placed equidistantly in a culture plate based on the seed size, and after completion of the incubation, the fungi growing out from the seeds on the culture medium, they are subjected for examination and identification purpose. It is based on the colony character, morphology of spores, and fruiting bodies under microscope. The beauty of this method is that the internally seed-borne fungi can also be observed through this method. Next, please. So this is a picture of agar plate method. You see, this is potato dextrose agar, and the second one is water agar. So how beautifully the colony is growing. The last one here, if you can see, one is showing black spots. and the fungal growth, a little fungal growth that is rather slow growing, but it has been observed in this water agar test because water agar doesn't contain any nutrient like potato dextrose agar. Next, please. Now modifications have been done. Hold fast method. I don't think it is keeping the time. It would be apt to describe all method details. Next, please. Glycol method is another modification of agar plate method that is specific to Magnoporthygrigia, paddy blast. Next, please. Advantages include that it is useful only for selective pathogens. Fruiting structures of fungi can develop quickly on agar, and the whole plate may be contaminated. The fungi that are not identified by Blotter method, that is internally seed-borne fungi, they can easily be identified by agar plate method because they come out of the seed. Pathogenic bacteria can also be detected because their colonies can easily grow on potato dextrose agar. Agar plate method provides adequate conditions for the mycelial growth, sporulation, and symptoms on seedling and seeds in a manner better than Blotter method. We can also isolate, purify, and identify the fungi that are found on seeds and can keep them in culture. Next, please. The limitations of agar plate method is that it is rather costly, laborious, and time consuming. It requires specific instruments and equipments. Slow growing fungi can be suppressed by fast growing ones, such as Fusarium and Sarcospora. They do not come, and other fungi like Rhizoctonia, they completely invade the whole of petri dish. Pathogenic potential of the associated organisms cannot be determined, as no disease symptoms may be produced in the poorly germinated seeds and weak seedlings on agar. Now, the analyst need next deep freezing blotter method. It is a modification of SMB method, as we discussed earlier. These after incubation at 20 degrees Celsius for a period of 48 hours, so that germination is induced. The plates are shifted to minus 20 degrees Celsius in a deep freezer for 24 hours. And they are again brought back to 20 degrees Celsius temperature under NUV and normal light for the rest of the incubation period of 3 to 5 days. It is recommended for detection of Fusarium and Septoria in cereals, Phoma in sugar beet, and Pyronophora gramineae and Pyronophora terrace in barley seeds. In this method, dead seeds act as natural substrate for fungal growth. Next. Now, this method has certain shortcomings like problem or overgrowth of saprophytes, rotting of seeds due to bacterial contamination after periods of freezing. Pathogens like Fusarium oxysporum and Fusarium moniliforme, which are rather saprophytic in nature, they either disappear altogether from the incubated seeds or change their normal growth character, so that to the extent that their identification under stereo binocular microscope becomes very much difficult, almost impossible. Some scientists have suggested that use of 8 hours duration of freezing at minus 20 degrees Celsius during incubation for better results. Next, please. This is the 2,4-D method. Next, please. Now, seedling symptom test. There are certain tests which are performed on the seedlings. Wiltner Big Stone method, sand method, standard soil method, test tube agar method. Because in seeds, all the pathogens and their propagules cannot be detected by a single method. So we need to grow seedlings to see that the seed-borne fungi which affect the pathogen in its seedling stage, they come on the seedlings only. So we need to grow the seedlings and observe them for the presence of fungi and diseases in them. So all these 4 methods are seedling symptom test. The symptoms are manifested on seedlings, and seedlings are raised by various methods. Next, please. This brick stone method, I think we are pretty much short of time. Next, please. Sand method, it's again needs some description. Next, please. Standard soil method, again seedling symptom test. Next, please. Test tube agar method. It's again the same that water agar or potato dextrose agar. Instead of petri dishes, it is kept on slants and seedlings are grown. 1 seed per slant is placed and seedling is grown and on the seedlings, because fungi, they are they readily grow on this PDA and water agar. Next, please. So it's extension of that. Next, please. Growing on test. It is used mostly in the greenhouse, polyhouse, or similar structures for protected cultivation. It's very important test for determining the viability of seed and detecting the pathogen that are seed transmitted. It is normally performed for detection and seed transmission of bacteria and viruses. Systemic infections can also be detected, and testing of seed by growing on test is generally accomplished by visual observation, indicator plants, serology, electron microscopy, or more advanced, more molecular biological technique. Next, please. Next, please. Growing on test is mostly, it is expensive, and it is done. It is more of quarantine significance. So governments, only government and higher budgets can afford it. Next, please. So advantages that these three advantages. Next. These are the limitations. Next, please. Hodge plague test. Next, please. Next. Let's move quickly to the serological test. ELISA, we all know ELISA is used for detection of many of the seed-borne pathogens. Next, please. Next, please. This method is described here, how it is performed, ELISA. Next, please. I hope you all might be aware of it. Immunofluorescence microscopy. It's rather an economic method, and it is used to detect or to confirm plant pathogens, bacteria, and seeds and plants. The antibody is bound to a fluorescent probe, usually fluorescent isothiocyanate, and stained bacterial cells can be visualized in an appropriately equipped fluorescence microscope. Next, please. The limitations of immunofluorescence microscopy. Next, please. Nucleic acid based detection method. There are certain techniques based on nucleic acid, such as PCR, okay, multiplex PCR, magnetic capture, hybridization PCR, bio PCR, many modifications of PCR. They can also be used for detection of many viruses, seed-borne viruses, bacteria, and fungi. Next, please. Next. The sensitivity level of nucleic acid based methods is very high. Previous slide, Dr. Can we have a comparison of the previous? Yes, this one. So this is a comparative analysis of the detection method. What kind of specificity or sensitivity, and what is the ease of application? How much time is required? All these 4 groups of assays: visual examination, microscopic examination, or seedling grow-out assay, serology-based detection, conventional DNA extraction, and PCR, nucleic acid-based method. Their sensitivity is very high, very high. So they are used to detect invasive and noxious pathogens and viruses while in quarantine. Next, please. So these are some newly developed diagnostic techniques. Biospeckle laser technique. It has recently been developed. Next, please. So videometer lab instrument. It has also been developed very recently, used to detect stem phylum Botryoideum, Cladosporium, Fusarium, Verticillium, Alternaria alternata. Next, please. Videometer lab. Now we are coming to the topic of seed protection. These were the methods, diagnostic methods for seed infection. Now protection. Protection is required for seeds from pests and diseases, weather damage, physical damage, nutrient deficiency, and genetic erosion. Next, please. There are many ways to protect seeds from these threats, biotic and abiotic threats. Some methods include seed treatment. Seeds are treated by chemicals or biological agents to protect them from pests, diseases, and other threats. Good storage practices, they protect seeds from abiotic aberrations, and they provide cool, dry place to protect them from moisture, heat, and pests. Biological control is also done. It involves using natural enemies of pests to control their populations. IPM is integration of a number of pest control measures applied to protect the seeds. Genetic engineering is also used to develop resistance of seed to pests and diseases. Protection of seeds is a complex issue, and we can say that no single solution can work for all the crops and all the situations. By using combination of methods, it is possible to reduce the risks to minimum and ensure the long-term viability and find the desirable results. Next, please. Some additional tips for protecting seeds include choose seeds from a reputable source that has good reputation for quality. Inspect seeds carefully before planting for signs of damage or infestation. At least visual observation can be performed at every level. Inspect seeds, sow seeds at the correct depth and spacing for optimum germination and emergence. Watering of seeds can be done regularly. Overwatering may be avoided, that may leave negative impact on seed germination. As per the need, seeds can be protected from pests and diseases with a variety of methods, including crop rotation, cover crops, and biological control. Next, please. Now, the improvement of establishment of healthy crops is significantly enhanced by seed production. And there are two types of techniques, conventional and contemporary techniques, that are used to protect seeds and store them. Next, please. What are traditional methods of seed production? Traditional methods, the seeds are coated with fine red soil, guntur chili, garlic powder, neem leaf powder, powder of bitter gourd seeds, drumstick extract, pongamia leaf extract, and they are stored for short duration. So these coatings, they protect seeds for certain period, for a very limited period, from invasion by pests and pathogens. Now, paddy seeds, they are immersed in salt water in the ratio of 1:10 to remove the floating chaffy seeds and then dried in shade for 1 to 2 years of storage. Sorghum seeds are treated with lime water, 1 kg of lime in 10 liters of water. It is S20, it is in fact S2O water for 10 days, and then seeds are dried and stored. Chickpea seeds, they can be treated with citronella leaf oil, cottonseed oil, soybean oil, castor seed oil at the rate of 500 ml oil for 100 kg of seeds. Sunflower seeds, they are kept inside the dried fruit of sponge gourd after removing the seeds. Seeds are removed from the sponge gourd fruit, and sunflower seeds are stored in that. These fruits were kept in an airtight container. Next, please. These are modern methods of seed protection. Seed treatment that we see in first photograph, seed protection, seed treatment. Second is seed pelleting, and the last one below, it's seed coating. So these all are the 5 methods: seed treatment, seed hardening, seed pelleting, coating, and biopriming of seeds. These methods are modern methods of seed protection. Next, please. Seed treatment, we will discuss one by one. In agriculture and horticulture, it is an age-old method. Seed dressing or seed treatment, it is a chemical, typically antimicrobial and antifungal, with which seeds are treated prior to planting. The benefits of seed treatment include that it prevents spread of plant disease, protects seeds from seedling blights, improves germination, seed germination and emergence, provides protection from storage insects, and controls soil insects. Next, please. It is an industry, seed treatment industry. What they do, that they have automated plants which require very little amount of chemical to treat a lot of seed, a very high amount of seeds. A very small quantity of plant protection products are required directly to the seed itself prior to the sowing. Next, please. Seed hardening is another technique of seed protection. Seeds are soaked in a chemical or water solution for a predetermined amount of time in the correct ratio. So that ratio is defined, chemical is defined. The seed is physiologically preconditioned by seed hardening, and it is then dried in the shade to its original moisture content and stored. The benefits of seed hardening is that it increases the yield, root growth, and vigor of seed germination. The uniformity of seedling emergence can be obtained, and flowering of hardened seeds occur 2 to 3 days earlier. So it is important and quite crucial. Uniform seed set can be obtained, and maturity time of maturity can be adjusted to simultaneous. It exposes the seed to drought tolerance also, because seeds, they germinate very quickly. They have been hardened and they germinate very quickly. Next, please. Now, these are the materials that are used for seed hardening. 1% potassium chloride, 1% potassium dihydrogen phosphate, 100 ppm succinic acid, 0.5% salt, sodium chloride, 100 ppm zinc sulfate, 100 ppm magnesium sulfate, 100 ppm ascorbic acid, 250 ppm cycocel, 0.5% magnesium sulfate, 2% calcium chloride, 1% or 2% potassium nitrate, 1% leaf extract of prosopis, pongamia, acacia, etc. And even ash. Ash can also be used for seed hardening. Next, please. These are the chemicals, hardening material for crops, different crops. 1% potassium chloride is recommended for rice and maize. 0.5% or 1% or 2% potassium dihydrogen phosphate is recommended for sorghum and pearl millet. 100 ppm succinic acid is recommended for seed hardening of cotton. 250 ppm cycocel is recommended for cotton. 2% calcium chloride is recommended for groundnut, and ash is recommended for most of the pulse crops, seeds of pulse crops. Next, please. So this is the standard. In rice, potassium chloride, 1% concentration and 10 hours of soaking. Sorghum, potassium dihydrogen phosphate or potassium chloride, 2% concentration and soaking time is 6 hours. Pearl millet, potassium chloride or sodium chloride, 2% concentration, 16 hours period of soaking. Finger millet, calcium chloride, 0.5% concentration and of the embryo growth. Maize, water, simple water is used for hardening, and 12 to 16 hours is period of soaking. Pigeon pea, zinc sulfate is used at the rate of 100 ppm and period of soaking is 4 hours. Next, please. It is seed pelleting. Now, enclosing the seed in a bioactive adhesive coated filter filler made of an inert substance. It is not filter, filler, but filler made of an inert substance is done. That is known as seed pelleting. It makes sowing faster and more efficient, and it can have a beneficial effect on germination. It also provides a secure and targeted way of applying nutrients and plant protection products directly to the seed. In some cases, seeds are pelleted using a rotary coater. Next, please. Next, Dr. Goyal. Protection during storage. Okay. Thank you. Protection of seeds during storage. These are while in storage, seeds need to be protected for the next cropping season, next sowing time. When the mother plant reaches physiologic maturity, storage begins right from there, right from that point. The preservation of the seeds from the moment they are harvested and collected until they are needed for the sowing is referred to as storage. Now, after harvest, these seeds are kept in warehouses, trucks, or retail stores for their protection from a number of biotic and abiotic factors that affect the seed quality. Next, please. Classification of seeds based on storage. There are three kinds of seeds: orthodox seeds, recalcitrant seeds, and intermediate seeds. We all know about them. Next, please. Awart divided seeds into three categories based on their longevity of storage. Microbiotic, which cannot be stored beyond 3 years. Mesobiotic, which the lifespan is 3 to 15 years. And macrobiotic, which can be stored safely for 15 years or more than 100 years. Okay, next, please. So, when we come to orthodox seeds, Harrington has laid down in 1908, he has laid down some thumb rules that for 1% decrease in moisture content, the storage life of the seed is doubled. And second rule is that for each 10 degree Fahrenheit, 5.6 degrees Celsius, that stands for decrease in storage temperature for the storage life of a seed is doubled again. And he has also coined the 100 rule, which indicates that the storage temperature in Fahrenheit and relative humidity in percent When we add them and it is less than 100, so the storage is safe. Later, it was viability equations were developed by Ely and Roberts. Next, please. Recalcitrant seeds, they cannot be stored very long because in deep freezing or in under lower temperature, the lethal ice crystals are formed during conventional storage, and they can kill the seeds. Next, please. So these are the methods of storage, conventional methods of storage, modern methods of storage. Modern methods include cryopreservation in gene bank, in Svalbard seed bank. Next, please. Conventional methods, we all know that they are stored in bamboo structures, earthen structures, wooden structure, underground structures in villages. Next, please. And improved ones include bins. Now, modern methods include in cryopreservation, storage of cells, tissues, embryo, or seeds. The germplasm is conserved by ultraviolet temperature in liquid nitrogen at 196 degrees Celsius. Minus 196 degrees Celsius is not practical for commercial seed storage because it is too much expensive. It is useful to store the valuable germplasm for use in the future, which cannot be preserved by conventional methods. Next, please. Now, seed storage gene bank. Next, please. This Svalbard seed bank. It's a very improved and modern method of seed storage. Seeds are stored in full price sealed envelopes, and then they are placed into plastic tote containers or metal shelving racks. Storage room temperature is minus 18 degrees Celsius. The low temperature and limited access to oxygen will ensure low metabolic activity and delayed seed aging. Permafrost means any ground that remains completely frozen for at least 2 years straight. This surrounding will help to maintain low temperature of the seed when the electricity supply fails. Next, please. Now, these are the challenges and prospects of seed health testing techniques and technology of seed production. Seed health, as we all know, that it is of paramount importance because without that, the term seed would be not be appropriate. They need to be protected from many of factors. And testing for health is important and gives an idea for developing strategy for their protection. Besides unique advantage offered by various seed and plant disease detection methods, every method has its own limitations, advantages and limitations. So none of these can be recommended for all kind of crops and all kinds of seeds. Before adopting these assays, it is critical to rigorously evaluate their applicability, precision, and accuracy of economies in real world. High throughput testing of naturally infested seeds. To ensure that these assays work, they must be validated in stringent multilaboratory tests, which evaluate their reproducibility and repeatability. Only assays evaluated in this manner should be considered for testing of commercial seeds. More research is needed to protect seeds under production and storage for ensuring the availability of quality seed. Next, please. Next. Dr. Goel?
Next.
Yeah. Now, we are at the end of our my presentation. I think I completed it within time. So this is our team. Next, please. There are some beautiful quotes before this slide. Next, please. So this is another quote. Robert Louis Stevenson. Don't judge. Yes. Don't judge each day with the harvest that you gain, but by the seeds that you are going to plant or you have planted. Next, please. So this is the summary. Protection of seed from a number of factors, both biotic and abiotic, is of paramount importance. Healthy seeds is prerequisite for a good crop stand and a remunerative harvest. It has also become an important quarantine issue, mainly in the international movement of seeds and germplasm exchange under seed trade. Thus, it is essential to make sure that no potentially damaging pathogens are established on seeds. The molecular methods of seed health testing have shown great potential for improving pathogen detection in seeds, as these embody many of the key characteristics, including specificity, sensitivity, rapidity, ease of implementation, interpretation, and applicability. Although they They are a little bit costlier, but they can be used very efficiently to in certain conditions. Methods for seed health testing, they lay down the guiding principle for the development of a suitable seed protection strategy. So this was the summary of today's discussion. Next, please. And now, thank you. Thank you all for patient listening. Thank you. Any questions
Thank
from
you, sir.
audience?
Any questions? Any questions? There's one question. Sir, can we say that a traditional method reduces the viability of seeder during storage in our Kashmir conditions? The farmers are relying on the method only. Asma, Dr. Asma is asking this question. Hello, Jaydeep sir.
Hello, hello. Yes, yes. I turned off my mic. So it is the chance is very rare because in Kashmir conditions, particularly in the valley, the temperature remains fairly below 25 degrees Celsius. Am I right? Hello.
Those days are gone, sir.
So temperature is increasing.
Now it is global warming, it is increasing. Nowadays, it goes up to 32, 34, even in Kashmir.
So depends upon the temperature, regional temperature, ambient temperature. If it is above 30, of course, the viability may be lost. But traditional methods are so economic that they are the only option left for the farmers